Hello, hi, a very happy good evening. So this is Krishna Ma'am and we'll be talking about the story of life on earth, and that's biology. So thank you so much for joining my session today. So please don't forget to like, share and subscribe. So we are continuing the respiratory pathways today. So we have started the respiratory pathways yesterday, and today we are continuing on the same. So yesterday we discussed about glycolysis in detail, and I hope glycolysis is no longer tough for you and it's pretty easy. So today we are going to the next one, that is the Krebs cycle. So before Krebs cycle we have the link reaction. So we're going to talk about these two today. All right. So let's go quickly. So respiratory pathways in plants. This is part 2. So let's see how much I have. So respiration in plants. So we started off with this, right? So I hope you remember. So I told you glycolysis is the most common process of all living organisms, the primary process that occurs in all organisms, right? So here the primary process is glycolysis, after wherein the glucose is broken down into pyruvate or the pyruvic acid, and then it follows the aerobic respiration separately and anaerobic respiration separately. So when it follows the aerobic respiration, right after glycolysis, it moves inside the link reaction or the Krebs cycle, and then it follows the ETS, that is the electron transport system. So when it is undergoing anaerobic respiration, so it follows your fermentation. So it can be either alcoholic fermentation or your lactic acid fermentation, right? Okay, so a quick recap about glycolysis. So what is that we were reading in glycolysis? So I'll just give you a quick recap. So glycolysis is a 10-step process. It happens in the cytoplasm of the cell. It was discovered by EMP, that is Emden, Mayer and Parnov. So it is also known as the EMP pathway. So here the substrate is your glucose, and it's broken down into a product that is pyruvate or your pyruvic acid, right? All right. So the 10 steps are as follows. So first of all, the glucose. So where does the glucose come from? The glucose is derived from sucrose, which comes from the part of photosynthesis. So photosynthesis gives you glucose. Right after photosynthesis produces glucose, this glucose is converted into sucrose. So this sucrose is how it is transported throughout the length of the plant, and this sucrose is broken down into glucose plus fructose using an enzyme invertase. Now this glucose and fructose enters your glycolytic cycle. So the first step being there is phosphorylation. So a phosphate group is getting added here. So glucose is getting an additional phosphate group at the 6th carbon. So from glucose to glucose 6 phosphate, it is getting converted. The enzyme that is used is hexokinase. Fine. So now the next step is glucose 6 phosphate is going to get converted into fructose 6 phosphate. So you know glucose and fructose are isomers. Yes. So when it gets converted into the isomeric form, so you have the enzyme isomerase, but since both are 6 carbon and both have phosphate, so it is phosphohexose isomerase. Okay. So next step is that fructose 6 phosphate gets an additional phosphate group. So that is fructose 1,6-bisphosphate. Bis is 2. So from one phosphate, it is getting the second phosphate. So that is fructose 1,6-bisphosphate. So here the enzyme is phosphofructokinase. So yesterday I was telling you, whenever there's a phosphorylation reaction, that is addition of phosphate, you have the involvement of the enzyme kinase, right? So this kinase is involved, and this is fructose on both the sides with a phosphate group, so it is phosphofructokinase. So wherever it is star marked, only those reactions are not reversible. All the other reactions of glycolysis are reversible. All right. So we go to the next equation. So this fructose 1,6-bisphosphate forms 2 molecules. One is DHAP, and it can form glyceraldehyde 3-phosphate. These 3 are reversible. DHAP can be converted into glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate can be converted into DHAP in the presence of an enzyme aldolase. So since both are interconvertible, so I conclude, or I take 2 molecules of glyceraldehyde 2-phosphate. 2 molecules because DHAP will also follow the same end. Glyceraldehyde 3-phosphate will also follow the same end. Right? So I have 2 molecules of G3P. So this 2 molecules of G3P gets converted into 2 molecules of 1,3-bisphosphoglyceraldehyde in the presence of glyceraldehyde phosphate dehydrogenase. So all you have to do is look into the substrate and the product and write the enzyme. Okay. So this 2 molecules of 1,3-bisphosphoglycerate, it becomes 3-phosphoglycerate, and then further 2-phosphoglycerate. Finally, it forms phosphoenol pyruvate, which in turn gets converted into your pyruvic acid or your pyruvate. So this is your entire step of glycolysis. right? So this is where we stopped. Then we started talking about the energy gain. So we are totally using 4 ATPs, we totally get 4 ATPs and we utilize 2 ATPs plus 2 NADH is being obtained. So the net gain is only 2 ATP plus 2 NADH. All right, yes. So today we move into the next reaction. So we are done with fermentation. So once this pyruvate is being formed, so its fate in aerobic as well as anaerobic reaction changes, right? So when you talk about in anaerobic reaction, right after pyruvate is formed, it gets converted into your alcohol or lactic acid based on whichever organism it is. If it is yeast, you have alcoholic fermentation happening. If it is lactic acid, so it forms your lactic acid bacteria, all right? Okay. So moving further, so today how it happens in aerobic respiration. So it is in link reaction. So right after, so it is a link between your glycolysis and your Krebs cycle. Okay. So how is the link happening? So I hope you remember the structure of mitochondria. So you have an outer membrane, you have an inner membrane, and you have the inner space, that is your matrix. So this cytoplasm of the mitochondria is matrix, whereas the cytoplasm of the chloroplast was called a stroma. So far when we are talking about the photosynthetic pathway, the C3 and the C4 that happened, we were dealing with respect to the chloroplast. But when we are talking about respiration, we'll deal with respect to your respiration, that is mitochondria, right? Yes? Okay. So pyruvate is transported from the cytoplasm to the mitochondrial matrix. Right after pyruvate is synthesized in the cytoplasm, it is quickly transported into the mitochondrial matrix. So once it is transported into the mitochondrial matrix, this is to show where your mitochondrial matrix is. It undergoes a process that is called as oxidative decarboxylation. That is the process it undergoes, and it gets converted to your acetyl-CoA. So 2 molecules of pyruvate get converted into 2 molecules of acetyl-CoA. Alright, and this process is catalyzed by an enzyme that is called as pyruvate dehydrogenase. So here we go. So this is what happens. So right after your pyruvate is synthesized in your cytoplasm, so this pyruvate is getting converted into acetyl-CoA. So if it's 2 molecules, you have 2 molecules of acetyl-CoA, but this is a decarboxylation reaction. When I say it is decarboxylation, you have the removal of carbon dioxide. So 1 carbon dioxide is coming out, right? So here 1 carbon dioxide is getting reduced, right? And this is catalyzed by an enzyme that is called as pyruvate dehydrogenase. But here you have to understand, so if it's for 2 molecules, so you have 2 molecules of pyruvate at the end of glycolysis. So you have 2 molecules of acetyl-CoA. So you have 2 molecules of NAD giving you 2 molecules of NADH plus 2 molecules of H+. Right? And this is catalyzed by the enzyme pyruvate dehydrogenase. Yes. So let's move forward. So this pyruvate dehydrogenase enzyme is a multi-subunit enzyme complex. So let's see how. So it has 4 coenzymes and it has 1 cofactor. So I hope with the revision of biomolecules, you remember what is a cofactor and a coenzyme. So a coenzyme is the non-protein part of the enzyme. So they are cofactors, right? So under cofactors, cofactors are the non-protein part of the enzyme. Under that, you have 3 categories. So you have prosthetic group, you have cofactors and coenzymes. So this particular enzyme of pyruvate dehydrogenase, so it has 4 coenzymes and it has 1 cofactor. So the coenzymes are TPP, thymine pyrophosphate, and it has lipoic acid, it has your coenzyme A, and it has your NAD+, right? And the cofactor here is Mg2+. So these are all the components that this enzyme pyruvate dehydrogenase is made of. All right? Okay. So now right after the link reaction, so this continues into the Krebs cycle. So what happens in the Krebs cycle? So that we are going to see. So he is Hans Krebs. So this Krebs cycle is named after the renowned scientist Hans Krebs, who elucidated this entire pathway. So he is Mr. Hans Krebs. All right. So it is also known as the tricarboxylic acid cycle. Okay. So it is also known as the citric acid cycle because the first stable compound that is formed is your citric acid here if you run the cycle. So these are the reasons why it is called so. So here we have how your citric acid cycle happens. So it is an 8-step reaction compared to your glycolysis, which was a 10-step reaction. This is pretty short. So let's see how easy it is. So first of all, your acetyl-CoA comes from your link reaction, right? So your acetyl-CoA is a 2C molecule, right? So this 2C molecule gets converted into citrate, that is 6C. So if it's one molecule of acetyl-CoA, so it will be one molecule of citrate only. If two molecules are there, 2 rounds of Krebs cycle will happen. Alright, okay. So this acetyl-CoA is getting condensed to form your citrate. So this is a type of condensation reaction where water is getting condensed to give you your SH group, so that your water is getting condensed, right? So your CoA will come out here. Okay, so this is what happens, the first step. So this happens in the presence of the enzyme citrate synthase. So your acetyl-CoA from the link reaction. So where does Krebs cycle happen? Krebs cycle happens in the mitochondrial matrix. All right, so acetyl-CoA gets converted into citrate by the process of condensation, which is catalyzed by your citrate synthase. Now this citrate will be converted into alpha-ketoglutarate acid. So this happens in the presence of isocitrate dehydrogenase enzyme. It is a type of decarboxylation reaction. If you see, citrate is 6 carbon and alpha ketoglutarate acid is 5 carbon. So 1 carbon is getting lost. So that comes out in the form of carbon dioxide. So it's a type of decarboxylation reaction. So decarboxylation, removal of 1 carbon dioxide. All right, yes, and here when this decarboxylation is happening, so here your NAD breaks down into NADH plus H plus. All right, so now your alpha ketoglutarate acid is broken down into succinyl-CoA, which is an intermediate step. This succinyl-CoA gets broken down into succinate, which is a 4-carbon compound. So this is a 5-carbon compound. So this again forms a 4-carbon compound here. So in the in-between step, so here it reduces, it is oxidative decarboxylation reaction. Again, a carbon dioxide comes out and NAD is oxidized to NADH plus H plus. Okay, so this succinyl-CoA is getting converted into succinate, and here you have the production of GTP. Okay. Alright, so now your succinate is getting converted into fumarate, alright. So in the presence of succinate dehydrogenase. Okay, so this is a dehydrogenation reaction, but here you have the production of FADH. So this fumarate is getting converted into malate. So this happens in the presence of an enzyme fumarase, which is a hydration reaction. Hydration is addition of water. Then the malate is getting converted into oxaloacetic acid, which is a 4-carbon compound. So this is in the presence of malate dehydrogenase enzyme. So everywhere there is 1 star, it means it is reversible. So the other star refers to the production of energy. If there are 2 stars, it means if there's 1 star, it means the reaction is reversible here. If there are 2 stars, along with the reversibility, there is also production of energy. So if you look into the Krebs cycle, which is very, very short, so you have 2 molecules of NADH, so 1 GTP, so GTP is equivalent to your ATP. So you have 1 more FADH and 1 more NADH, right? So let's calculate. So this is how your entire Krebs cycle happens. So we have totally, how many NADH we had? So 1, 2, and in the next slide we have 1. So totally for 1 molecule of acetyl-CoA, so for 1 molecule of acetyl-CoA, you have 3 NADH plus 3H plus. You have 1 molecule of GTP, which is also equivalent to your ATP. And then you have 1 molecule of FADH. Plus you can write H plus if you want. So this is for one molecule of acetyl-CoA. But at the end of glycolysis, you have two molecules of acetyl-CoA. So for two molecules, how many will you have? So you will have 6 NADH plus 2 GATP plus 2 FADH. Right? So this is the net gain from your Krebs cycle. So moving further, we are going to talk about the enzymes. So all the enzymes for your TCA, so we spoke about 8 enzymes, right? So all the 8 enzymes are present in the matrix of the mitochondria except one. So that one is your succinate dehydrogenase. It is present in the inner mitochondrial membrane. So this is the only exception that I want you to remember. But so here we'll see. So in glycolysis, what was your gain? So your gain was 2 ATP plus 2 NADH. So now talking about your link reaction. So for two molecules of pyruvate or pyruvic acid, so you have 2 molecules of NADH. So then talking about the Krebs cycle, so for 2 molecules of acetyl-CoA, how many we have? So we had 6 NADH plus 2 ATP plus 2 FADH. So for one molecule of glucose, you have so much of energy. So we'll count all the ATPs. So we have 4 ATPs. So we have 10 NADH and we have 2 FADH. So this is the amount of energy that is obtained from the respiration or the oxidation of one molecule of glucose. All right. Yes. So, yes, ma'am. A very happy good evening. So thank you so much for joining my lecture today. So please do like, share and subscribe. So I hope this is clear. So with this, we are done with the second part of your respiratory pathways, wherein we are covered with your link reaction and Krebs cycle. So on Monday, we'll be talking about your ETC. So that will be the last portion of your respiratory pathways, and then we'll move on into human physiology. So a quick highlight of human physiology will be done. All right. Yes. So if you genuinely like my way of teaching, so please do like, share and subscribe, and please let me know your comments in the comment section. So I hope you are benefiting from these sessions. So I'll meet you on Monday at 5:30. Sorry. So the class timing has been changed. So Monday we have a class at 5:30. So on Monday at 5:30 I'll meet you. So until then, this is me signing off. So take care and bye. Thank you.